basic code for life on Earth is DNA. Scientists once believed the biological cause of all traits was encoded in the DNA sequence. This prompted the Human Genome Project, an international research effort to sequence and map all of the genes, together known as the genome. On June 26, 2000, as researchers announced the completion of sequencing, United States President Bill Clinton said, "This is, without a doubt, the most important, most wondrous map ever produced by humankind. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases." Given this impressive accomplishment, it is surprising that the precise cause of genetically related illnesses and diseases aren't known. Unfortunately, unraveling the sequence of DNA is not enough to understand all of the nuances of life. A burgeoning field of study, known as epigenetics, aims to explain the natural phenomenon which conventional genetics has so far been unable to answer. Conventional genetics was guided by a simple principle. One gene, the functional unit of DNA, encoded one RNA, which encoded one protein, which produced one biological trait, known as a phenotype. These phenotypes did not change unless the associated DNA sequence changed as well. Biologists now know that this view of genetics is oversimplified. Numerous processes affect when, where, and to what extent genes are expressed in a cell. These epigenetic phenomena are all around us, and all meet one of two simple criteria. 1. Two organisms are genotypically identical and phenotypically variable, like identical twins, born with identical genomes, who grow up into two very different adults. Or 2. An organism continues to be influenced by an event long after the initiating event has occurred, like how a single-celled zygote is exposed to distinct hormone signals, which then turn that cell into numerous distinct adult cell types. These environmental signals, in this case developmental hormones, produce permanent changes in the cell, and these changes are heritable, as they are passed on to daughter cells. Though many epigenetic questions scientists have pertain to humans, they rarely use humans in research, as this is both difficult and time-consuming. Therefore, many researchers use model organisms instead.
Our lab uses the fission yeast, Schizosaccharomyces palmbae, as a model system to study epigenetics because it's easy to manipulate in the lab and because it shares many molecular features with humans. It can survive being frozen, so we keep various stocks of mutant strains readily accessible. When needed, we thaw a mutant, and it quickly rejuvenates. Palmbae's short generation time of 2 to 4 hours means that within 3 days in an incubator, we have complete regrowth of the yeast. With a little more time and space, Palmbae can be safely grown in bulk. This 1-liter lab-grown yeast culture contains about 20 billion microorganisms, yet is as safe as your average beer.
Not only are they convenient and safe, but fission yeast shares many chromosomal structures and features with humans, meaning that findings in yeast often correlate to findings in humans. Fission yeast are eukaryotes with 3 large chromosomes, with large sections of epigenetically silenced DNA domains, vital to any study of epigenetics. The fission yeast life cycle contains both haploid and diploid states, which makes manipulating the genome easy for epigenetic studies. Haploid organisms only have one copy of each gene within a cell, so the removal or knockout of a gene when they are in the haploid state produces full mutants. This is much more efficient than attempting to create mutants in diploid organisms, where both copies of a gene must be mutated to create a full mutant. Palmbae can also produce diploid cells through sexual reproduction, meaning 2 mutant strains can be crossed to produce a strain with multiple mutations. Epigenetics is complex to study, as it is a phenomenon that occurs at the boundary between biological determinism and a lifetime of experiences. We use fission yeast because they speed up the research and allow findings to more quickly move into implications for human health research.